Tonight, we're privileged to hear Dr. Rob Carter. He's going to be speaking on ancient DNA. Dr. Carter obtained his Bachelor of Science at Georgia Institute of Technology. Then he taught some high school biology, chemistry, physics for 4 years, and went back and obtained his PhD at the University of Miami in marine biology. And he's currently researching genetics, human DNA, and speaks for Creation Ministries International. He also has a number of resources available at CMI, Creation Ministries International. So we're excited to hear what he has to say about the origins of human DNA and how that may correlate with the Genesis record of human existence. Before we begin, let's offer a word of prayer. Dear Creator God, Heavenly Father, and dear Lord Jesus, we thank you for the blessings you give each of us. We thank you for the miracles that surround us, that point to your creation of this marvelous universe, everything in it. And we thank you for the miracle of the gift of life in every living thing. Now open our minds and our hearts to the message that Dr. Carter presents tonight. Increase our understanding of your incredible works in this world around us. In Jesus' name we pray, amen. So, Dr. Carter.
Well, hello, everybody. I cannot see anyone there, so I'm going to assume that there's a huge audience just saying, rah, rah, go, go. No, I'm just kidding. My name is Robert Carter. I am a speaker for Creation Ministries International. You are one of probably about 12 or 1,300 groups that we're going to speak to this year somewhere around the world. There are about 30 people like me on the speaking circuit. We each have our different expertises. And one of my expertises is in genetics, and I love studying DNA. Now, you heard that I got a PhD in marine biology, but it's really in coral reef ecology. And honestly, all my work is done in the genetics lab, sequencing DNA, taking DNA out of corals and putting it into bacteria, taking it out of the bacteria and putting it into fish, making transgenic fish. And that's really what I've been doing now for about 25 years. I love studying DNA, but DNA gives us the ability to test theories of history, because part of genetics is a transfer of DNA from one generation to the next, to the next, to the next. And if you look at enough DNA, you can actually start looking backwards in time and maybe making some educated guesses on past events. Well, the Bible claims to be a history book, and genetics is gives us some tools of history. Can we test the Bible using genetics? Actually, we can, and that's the subject of several of my other talks, but this one, we're going to go way, way back, as far as we can possibly go in genetics, and look at ancient DNA. That is, DNA that they're able to pull up from old bones and now even cave sediments in different places around the world. And we're going to see that it's actually telling a very biblical story, unlike what the evolutionists want us to believe. Now, does this work? It does not. Okay, I'm going to do... how do I do that? Click, touch there. There we go. Okay. If you look in the Bible, we're given a lot of information about where people came from. We're given information about entire cultural groups, family lines, long, detailed genealogical lists. In fact, the Bible quite clearly claims that all people in the world that exist today came from Noah and his family, maybe 4,500, maybe 4,600 years ago. That's not a long time to explain where, Africans and Asians and Native Americans and Europeans came from. Now, I think we can definitely do it, but it is not a long time. And we've got to somehow take all this information we see in people and put them into Noah and his family. And that's a bit of a challenge. One thing, though, that happens across the world is most people in history married someone from their own group. There's a lot of inbreeding amongst tightly related cultural groups. We see it here, and what I'm describing on the board here is a family tree of the 12 tribes of Israel down on the bottom. And in blue, those are all the people that we know contributed to the 12 tribes of Israel. And when you look at the top where Terah is, you realize that Abraham and Sarah are half brother and half sister, and their son Isaac married his first cousin once removed and first cousin twice removed, Rebecca, and their son Jacob married his first cousins Rachel and Leah, who are much closer than first cousins because they're related together on multiple different branches of the family tree. You realize there's a lot of inbreeding in that family. What that does, though, when we look at old cemeteries, you can see family groups, you can see cultural relatedness. There's one point where these people are migrating into Europe. It's the first hunter gatherers that are moving in. No, that's not true. It's the farmers that are moving in. And the farmers came up the Danube from the west, and they went across the Mediterranean, came up through like Spain and France. And when they met in Central Europe and like Germany, you could genetically tell the difference between the two groups because they'd inbred along the path so much, they're very different from one another. But we know that because of the DNA that's in the graves. But they were living right next to the hunter gatherer people. Now, to be a hunter gatherer, I don't want to insult anyone. That doesn't mean stupid. In fact, you got to be really smart to live off the land and not have farms. You have to know the climate cycles, you have to know the migration patterns, you have to know where to find the mushrooms and the berries, or you're going to die because the weather can be really unforgiving, of course. But we have multiple cultural groups for living right there in Europe, and we can look at DNA, we see a very complicated pattern. You okay with that? Because that's the reality of the data on the ground. Our challenge is we got to fit all of this history into a short time frame. If we look at just a biblical timeline of history, you got creation about 4000 BC, a little more than that. The flood's about 1600 BC or 1600 years after creation. That is, the Abraham lived right about 2000 BC. Moses lived right about 1500 BC. David literally lived at 1000 BC. Of course, Jesus came at year zero, even though that doesn't exist, and today we're 2,000 years after Jesus. So that's our time frame, and we've got to take Neanderthals, we've got to take Homo erectus, which we don't have any DNA from yet. We've got to get the hunter-gatherers in Europe and the early farmers in Europe. We've got to get the people who invented potatoes in South America many thousands of years ago, and then invented corn and bred these brand new crop things. I mean, these are massive civilizations that are popping up in South and Central America that we've got to stick onto a very short timeline. So acknowledge that this is a challenge, and I think it can be done, of course, because I wouldn't mention it. I think if it couldn't be done, but we can definitely do it. We can also put the so-called ages onto the timeline. You've probably heard of the phrase Bronze Age. Well, how long was the Bronze Age? When I hear age, I think thousands of years. But the Bronze Age was a few hundred years. In fact, some places didn't even have a Bronze Age. They went straight to the Iron Age. Some places the Bronze Age was longer and the Iron Age came in very late. Sometimes the Iron Age started early. So it all depends on what you're talking about. And you can't actually take a Bronze Age China and compare it to Bronze Age Israel. They're not comparable. Those are different Bronze Ages, different metallurgical, whatever, different developments of metal technologies in different places at different times. So it's kind of hard to fit all that into the Bible until you realize that these ages are really short. So Moses lived in a Bronze Age. David lived in, I think, it was Iron Age 2A, I think. So not just the Iron Age, but a subgroup of the Iron Age timeline. That's when David lived. Yeah, the Battle of Troy, that's in Homer, was during the time of the Judges. Oh, wait, if the Iliad and Troy and all that was the time of the Judges, then all of Greek history fits into the biblical timeline very easily. In fact, that's true. The Peloponnesian Wars were 400 or 500 BC. Oh, I thought that stuff was ancient. No, it's not really that ancient. Consider Genesis chapter 9, verses 18 and 19. It says something very genetic. The sons of Noah who went forth from the ark were Shem, Ham, and Japheth. These three were the sons of Noah, and from these the people of the whole earth were dispersed. All right, Christian, answer this question. Does the Bible claim that all people on earth today descend from Shem, Ham, and Japheth, and Shem, Ham, and Japheth alone? Yeah, it does. That means that we've got to get all the genetics of all modern people and all ancient people onto the ark. Yeah, it can be done, definitely, but that is what is required if this is true. This means that the flood is global, not local. It means that all people go back to, all Y chromosomes go back to Noah. We don't actually have a Y chromosome Adam because we have Noah. He's the Y chromosome Adam. So the evolutionists should have called him Y chromosome Noah, but they didn't. Why? Because they were biblically ignorant. Just saying. And we have this thing in Genesis chapters 10, 11, we call the Table of Nations. We have this account of all these people groups descending from Noah. And we have a lot of named people, but then we have groups like the Hamathites on the bottom right, or the Kazluhites in the middle on the right, or the a lot of things that end in eem. Eem, of course, is the Hebrew plural. If you have an eem, that means more than one thing. An ite means a descendant of or a citizen of. So we have Noah, Shem, Ham, and Japheth, I think 16 grandsons, some great grandsons, but most of the greats are people groups. And you wouldn't expect necessarily, like say the Kapthorim. Kapthor is the ancient name for Crete. So the descendants of Ham actually lived on a European island called Crete. But just because the Kapthorim descended from Ham doesn't mean every single person in that culture was a Y chromosome descendant of Ham. Because when you have an island, you have trade routes, you have slavery, you have warfare, you've got people moving around. And when you have any kind of an established city, you're going to get people migrating in toward that city and you get a mixing. So, yeah, the Kazluhites, the Kapthorites, they descended from Ham, but it doesn't mean that's exclusive. We have to understand that because that's going to influence our views on genetics. Now, we've already established for Europe specifically that Shem, Ham, and Japheth all had descendants in Turkey, and the Turkish people, not Turks yet, but the Anatolian people, are going to migrate into Europe en masse, and that's going to have a profound effect on the genetics. I know that in the creations community, there are a lot of people that struggle with ancient DNA. We're going to get into some of the technology in a minute, but there's a resistance to trusting it. Because how can you trust DNA if it's all broken down? Because DNA in the ground, it breaks down, it falls apart, it's chemically reactive. It doesn't. When you pull when we sequence the human genome, we pull out gigantic long pieces of the human genome. Because our chromosome 1 is a foot long. That's how long one molecule is in our cells, and you can just sequence that for a long, long way before for the sequencing machines that we have. But when they sequence the Neanderthal genome, the longest piece they found was about 50 letters. It's highly fragmented. So how can you trust that? I mean, how do you know that's actually Neanderthal? And how do you know it's not just a whole bunch of mistakes? Well, the reason for that is we have more than one Neanderthal genome. In fact, we have many now. In fact, a couple of high quality Neanderthal genomes, and they keep giving us the same answer. When you pull a DNA, oh, it's Neanderthal DNA, not modern human DNA. Therefore, since we can do it many times and we keep getting the same answer, that really is an accurate representation of the Neanderthal genome. We'll get back to Neanderthals in a minute. But knowing that there's some skepticism, and we have room for healthy skepticism, fine. But we can ask the question, how do you get ancient DNA and how can you trust it? Well, the first time anyone tried extracting DNA from an extinct organism, no one thought it was going to actually happen. It was a test. They went to a museum specimen of a quagga that went extinct. I think it was the quagga that died in 1883, the last one alive, and they pulled DNA out of the skin, and they're like, "What? There's DNA in that thing? That thing died, over 100 years ago. How can there be DNA in it?" Wait, how far back can we go? And so they started pulling older and older and older specimens. They kept going back further and further. And yeah, the DNA was decayed, but it was still there. And then they said, okay, really, how far back can we go? And we've now been able to sequence the genomes, I mean, high quality genomes of ancient horses, cave bears, mammoths, mastodons. Now, notice those things sound ice age ish. Well, there's a reason for that. That is, the most of the ancient DNA specimens we have are from high latitudes where it's cold. And that cold is important because the low temperature helps preserve the DNA. We have a really hard time finding DNA in Africa. There's almost no ancient DNA. There's a little bit, but not hardly any. And that's a problem because that's, the hotbed of human evolutionary stories. And we sure would like to get some more ancient DNA from there. We do have a little bit. But most of it comes from Ice Age type things. The other reason it's Ice Age is because the evolutionists didn't expect to find DNA in something from, let's say, that they date 1 million years ago, or 2 million, or 3 million years ago. And so they didn't look. And then when they started looking, they started finding DNA there also. But one of the coolest things that's happened was the realization that the inner ear bone, it's called the petrous bone or the rock bone. The petrous bone can contain sometimes 100 times more DNA than any other bone in the body. And because it's so dense, the DNA inside is protected from water and oxygen and it resists decay. And once they realized that, they're able to go back to some old skulls and just drill a little bit of material out of the petrous bone and sequence a fairly high quality genome from a lot of extinct individuals. And that was amazing. But DNA in the ground does react very quickly. It does not last long. One problem that happens is called cross-linking. If DNA touches itself, if the two strands cross, they can actually react with each other and form a crosslink. And then you have a DNA circle, and a sequencing machine can't sequence that because it gets to that crosslink, and the enzyme that's doing the DNA sequencing is blocked and has to stop. So crosslink DNA is really hard to sequence. In fact, we had a hard time sequencing DNA out of mummies from ancient Egypt because of cross-linking, because of decay, and because of the resins that they used in the embalming process. It took us a long time to figure out how to get around those things. And now we don't just have DNA, we have DNA of King Tut and his mother and his grandparents and his wife and a whole bunch of pharaonic uh, mummies from different places in the Egyptian history. And I can't wait for them to fill out that family tree even more. But we can definitely pull DNA out of those things. Something else that happens to DNA in the ground, very, very common, is called cytosine deamination. DNA reacts with water. This happens in your cells also. Is one of the main things that contributes to mutation accumulation over time in the human population is the transition of a C to a T. That's cytosine to thymine. So when cytosine reacts with water, that NH2, that amine, get ripped off and replaced with an oxygen. But now we have a molecule called uracil, and uracil doesn't belong in DNA. It's in RNA. So in our bodies, we have very complex enzyme systems that will find the uracil, cut it out, and stick the C back in there again. Sometimes it makes a mistake and it goes the wrong direction, and therefore you get a C to T. But in dead people and dead remains of animals also, the C becomes uracil and stops because there's no enzymatic system to fix it. And so you have DNA that's filled with uracil. And sequencing machines have a hard time sequencing that. So one thing they do is they treat it and they flip it back to cytosine. Or when they sequence it, they look for the C to use in the sequence DNA and they trim off the end of the DNA that has lots of U's because DNA tends to react at the ends. So even if it breaks, it'll start degrading from the ends, and the central part tends to be not mutated or not chemically reacted. So they sequence a piece of ancient DNA, cut the ends off, and keep the middle. If you look at DNA, and I'm hoping you don't hear the sound of this video is playing. We tested it earlier; they didn't hear it, but I'm going to talk over myself if we did. If you look at DNA in the cell, it breaks down really quickly. If you look at DNA in the ground, it breaks down even more quickly. Talking about double strand breaks, single strand breaks, C to T, C to U that is, and all sorts of other things. DNA is an incredibly unstable molecule. And so it's a massive problem when we're trying to study ancient DNA. In fact, some studies on ancient moa bones found in New Zealand, they were able to look at the half-life of DNA of different lengths and at different temperatures, and they showed that even DNA of any length is going to break down into very small pieces very quickly. In fact, it doesn't take too long before you get complete decay of the entire DNA chain where you don't even have one letter stuck to another. But way before that, it's unsequenceable. You need a long piece of DNA to be able to sequence it. If you've got something that's 5, 10 letters long, it's worthless. You cannot sequence it. So as far as applying sequencing technology to ancient DNA, you need pretty good DNA, but it breaks down way too fast. And it was such a disappointment for them. This is one reason why they didn't expect to find any ancient DNA in old bones. But something else very important to understand about this. This was based on carbon dating. They dated the Moa bones with carbon 14. And there's biblical reasons to think that carbon 14 will have been, there'll be less carbon 14 in the world before the flood. And as Earth's magnetic field decays and carbon 14 builds up, you have an equilibrium rate today, which is much higher than in the past. Well, since carbon 14 looks at how much carbon 14 is present, if you had less carbon 14 in the past, anything you're studying will be artificially aged. It'll look older than it really is because it has less carbon 14 than the evolutionist expects. So these DNA decay rates are actually faster than this. The DNA decay rates they measured were too slow because the Moa bones aren't as old as they think. And if the Moa bones are younger, then the DNA has to decay faster to get these results. This is a massive problem. Think about this. We find DNA in things that they claim are hundreds, thousands, or millions of years old. That's not possible. The very presence of ancient DNA tells us that the world is not as old as they say. I published a putative DNA or carbon 14 decay curve in an article called How Carbon Dating Works. I really like this article. If you ever wonder about carbon dating, how the machine works, how the calibrations work, and how do you apply it to biblical history, that might be a good resource for you. How Carbon Dating Works on creation.com. Well, what I reasoned was that as you go back in time, the carbon dates are going to go much higher in the past until you get to the antediluvian level. And so when we carbon date dinosaur bones, they typically come out 56, 60,000 years old. That's a very typical dinosaur carbon 14 date. It doesn't mean it's that old. It means the pre-flood world didn't have much carbon 14, but it did have a little bit, and that's what we see in the dinosaur bones. Carbon 14 affects our understanding of ancient DNA and ancient DNA decay rates. It's very important to know. So how do they do it? Well, first of all, they find something that they want to pull DNA out of, and they wear a mask and they wear gloves, and they do it in a sterile environment. When Lee Berger found those tiny little hominids in that cave in South Africa, the Rising Star Cave, this is Homo naledi, they didn't wear gloves when they were handling the specimens. And I just slapped my head. I was like, how can anyone not know? How can any scientist today touch an old bone with their hands when you have DNases in the oils of your skin? As soon as you touch a bone, you have contaminated it. So they didn't do that. People who are looking at ancient DNA, though, they will do clean room technology, and then they'll just pull all the DNA out of a sample, and it's all broken down DNA, all decayed DNA. Fine, they just pull it all out, and then they'll mix basically with like a barcode. They have a sequence of DNA that they made in the machine, and it's all random sequences, and they'll tag the DNA by enzymatically joining a tag to every piece of DNA. So every piece of DNA in the test tube has its own unique identifier. And then they do what's called the polymerase chain reaction. They use bacterial enzymes to make copies of that DNA. And then they run through a cycle again, they make copies. And they run through a cycle again, they make copies. And so what you got, you go from like 2 to 4 to 8 to 16 to 32 to 64. You have an exponentially growing amount of DNA in your test tube. And then they take that and they throw it into a sequencing machine. I'm not going to explain how the sequencing machine works. They are brilliant, especially the most modern ones are amazing, but they will sequence the letters of that DNA. So you get A, C, C, G, C, C, T, 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 A, E, C, 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 G, G, T, T, whatever. And they'll look at that, and then they have to apply some human quality control. Here's where the art comes in, and here's where some of the evolutionary biases can creep in very easily. They will take every long sequence and throw it out. Because, as we know, DNA breaks down very quickly, so anything that's long must be modern DNA. Bacteria, fungi, human contamination, they'll just throw it out. And then they'll look for um, things with a lot of uracil. So, okay, that must be ancient DNA. They'll take that. Then so you have the short piece of DNA that's highly degraded. That's the definition of ancient DNA. They chop off the ends. They take that little part in the middle, might be 20 letters, maybe, and then they have to align it. We could not have made the Neanderthal genome if we didn't have a human genome. Because to build the human genome, you had to sequence millions of pieces, like 300 letters long that the machines back then were doing, and then line up the pieces. So if this has AAAG, this has AAAG, okay, they line up. And then if you take millions of pieces and line them up and stack them on top of each other, you can get a pretty good estimate of what the human genome is. You can't do that with fragmentary DNA. So what they did is they took the pieces of not chimpanzee, of Neanderthal DNA and searched the human genome for that piece. That's where that piece goes. And they're able to assemble about 60% of the Neanderthal genome doing that. That's really interesting. That's the technology. That's how they do ancient DNA. Now, let's look at some case studies. Some things that you're familiar with. Let's start with Native Americans. When we look at the DNA of Native Americans, it is clear that they came from people living in Asia, but not the people in Eastern Asia, not Chinese people. They're actually related more closely to people from Central Siberia. So it looks like a migrating band of people split, and some of them crossed into the Americas and became Native Americans. But one estimate said maybe 70 people crossed. That's it. And because of their low genetic diversity, that's why they had such massive problems when Europeans arrived. Because Europeans have a much higher genetic diversity. So if a bug figured out how to kill one European, it might not necessarily kill another. But you get a new virus amongst Native Americans, which are very homogenous. And once a virus figures out how to kill one, well, it could probably kill 95% of them. So I don't know what percent of Native American population died after first contact. I suspect it was over 95%. I'm guessing maybe 98%. Nobody really knows, but I think the low ball estimates are underestimating the true death rate. So a couple hundred years later, when waves of Europeans are coming in, they're finding this vast empty area. Well, yeah, it looked like a wilderness because the people that were surviving were living basically a post-apocalyptic wasteland. Their culture had been destroyed. But really interestingly, the earliest samples of Native American DNA that we found are similar to the people that live here today. There was a famous case of Kennewick Man. Now, Kennewick, Washington, if you're coming across from Siberia and across through Alaska and following the coastline, when you get to Washington, you're going down the coast and you can, islands and things like that, but you can't get inland because there's a range of mountains. But when you get to the big river, my brain's hiccuping a moment. Yeah, you're all shouting at me right now. The name of the river. Thank you. You take the big river inland. You can go all the way to Idaho, and the first major settling area is Kennewick, Washington. So it's really the gateway to the inside of the United States. And some scientists dug up a skeleton. And they said, oh, he's a European. Well, here's an ancient skeleton with European features. All the conspiracy theories came out, all the alternate history people came out, and Native Americans said, no, that's ours. That's one of our ancestors. How dare you dig up our graves? Give it back. And the scientists said, no, he's European. They said, no, he's not. He's ours. And so it was a lawsuit. And they did a compromise. They said, okay, look, let us take DNA and sequence the DNA from this individual. If he's Native American, we'll give him back. They didn't think he was going to be Native American, and he was. Not only that, he actually was closely related to the tribes living in that area. So the Native Americans that got there a long, long time ago have lived there right there the whole time. And yeah, someone was digging up someone's great, great, great something grandfather, but that was Kennewick man. That was a very overly brief description of the genetic status of Native Americans. Now let's go to East Asians. Let's go like to China. Really interesting. There was they dated 40,000 years ago, 40 KYA. They said a modern human from a cave near Beijing. They found him, they sequenced his DNA, and his DNA was very similar to the people living in the area today. Again, genetic continuity, but their timeline is messed up because of carbon dating. It's way too early. It's not real, but there is continuity over at least thousands of years in many places in the world. And again, the Chinese people are less related to Native Americans than the Siberian people living to the west of China. Okay, now let's go to the west of Eurasia and talk about Europeans. We are talking about Japheth. No, Europeans did not descend from Japheth. Strangely, and this has been consistent from all the samples, maybe 30, maybe 50 now, um, genomes of ancient Europeans, the ancient most Europeans. First, we have the Neanderthals, then we have these hunter gatherer peoples, and their genomes tell us that they had dark skin and blue eyes. At least they carried the genes, and when those genes appear in modern people, it causes dark skin or blue eyes. Now, dark skin and blue eyes is a rare combination today, but all of the ancient Europeans had it. At least the ancient modern Europeans had it. That's strange. We have multiple levels of archaeology in Europe, and each one is associated with a different genomic context, waves of people migrating in. They did something amazing in Denmark. They found a piece of birch tar that had been chewed. It had human teeth marks in a bog, and they said, "Oh, well, this is in a oxygen-free, acidic environment. That's a perfect place to preserve DNA." So they took this piece of birch tar that had been chewed, and they pulled the DNA out of it, and they found, guess what? Birch DNA and duck DNA and hazelnut DNA and human DNA. So this is a Neolithic woman whose last meal was duck and hazelnuts. She had the genes for dark skin and blue eyes, and she was living in Denmark. That is so cool. But it's a curveball. It's not something we expected from the biblical picture. And our very simplified versions of ancient history, well, they're wrong. We need to get rid of those simplified ideas and understand that human history is a lot more complicated than the Sunday school version of the book of Genesis would teach us. Now, later on, we got the hunter gatherers up to the evolutionary dates of 35,000 years ago. And then the early European farmers didn't move into Europe until fairly late. They say 9,000 years ago. That's not a long time ago, especially if you compress the dates to put it into biblical history. But this is the first time when the SLC24A5 gene comes in. That's the gene for light skin. It came from Anatolia, ancient Turkey. In fact, if you've heard of Otzi, the Tyrolean Iceman that they found on the border of Austria and Italy under a glacier as the glacier melted back, I mean, it's amazing, amazing find. Well, he had that gene, and he was an early Neolithic farmer. These people are moving into Europe, living side by side with the Western hunter-gatherers. Interestingly, the Western hunter-gatherer DNA bleeds into the farmer's DNA, but not vice versa. As far as some sites in Brittany, they find these Western hunter gatherers that have no evidence of interbreeding with the farmers, but over in the farming territories, you have farmers with Western hunter gatherer DNA. So there's very complex cultural interactions happening here as these people are meeting each other. But then something even weirder happens. You might think, okay, fine, we just described Europe. No, we haven't. That's maybe 30% of the European genome we talked about. The other 70% is still a mystery. You dig up a person living in Germany or France or England from a few thousand years ago, they don't have the same genome that you and I do. And we're looking in vain. Where are these people living? Where are the Europeans? Well, they found them out in the steppes, the grasslands north of the Caspian Sea. If you've ever seen Fiddler on the Roof, yeah, that area, the vast rolling plains that stretch for thousands of miles across Asia, that's where the Europeans were living for a long time. And then they swept westward and they swept away and they intermarried with, but their culture came to dominate. It came from this. If you're European, this is your homeland. It's not France, it's Kazakhstan. Back to the Yamnaya cultural horizon, they call it. These people swept eastward and westward. We knew that from the archaeology. We knew that from linguistics, which is why Hindi has so many root words in common with Italian. Said, wow, there must have been a culture. Now we know it from the DNA as well. Europeans, that's where you came from. I know it's strange, but that's the genetic evidence. But putting all that together, though, we have to understand that the concept of race from a genetic standpoint has been absolutely destroyed. This is a quote from Slatkin and Racimo from the Proceedings of the National Academy of Sciences. They wrote, "We now know," I love that phrase. When a scientist says we now know, that means we were wrong yesterday. Put that in context. We now know that present day populations were created by a complex history of admixture and population movement. Although local genetic continuity over long periods has been documented in a few cases, these are exceptional. The general rule is that the ancestors of present day populations lived somewhere else. And we can see that all over the place, specifically in this case, in the Y chromosome family tree. Now, I don't have the ability, I think, to turn on a laser pointer here, do I? I got a pointer, okay, I don't know. Boom. Maybe you can see my pointer. I don't know if you can or not. But when we look at the Y chromosome family tree, now I made this chart. I took several hundred human Y chromosomes. I put them into evolutionary tree building program to build this. We have the Khoisan Bushmen that live in the Kalahari Desert are group A. Then we have the pygmies that live in the Central African rainforest. That's group B. Then we have E1B1A. That's 95% of African males today. But they branch off from E1B1B, which are North African and Middle Eastern, but they branch off from E and D, and Genghis Khan was group C, the Mongolian warlord. So even the evolutionists are admitting now that this group E1B1A, that's most of the African men, did not arise inside Africa. Because that branches off from non-Africans, and that combined group branches off from non-Africans, etc. They arose outside of Africa and then invaded and took over. And if you look at these, like these look like broomsticks. We have a tuft of bristles, and then we have a long stick. Well, that tuft of bristles goes back to a single man who's an ancestor of 95% of African men. And the length of the branch is proportional to time, not exactly equal to time, but it's proportional to time. So this African group arose fairly recently. Oh. Well, I said before, I'm R1B, that's my group, but also look, it's bristles that go back to a common ancestor. And the common parallel group is R1A. Those are people who live in Pakistan and India. Oh yeah, that's the Indo-European language group. Oh, and it's also reflected in the archaeology and the genetics. Oh, that's really cool. Group Q is Native Americans. Oh, so they're more closely related to Europeans than Group O, which are Chinese, Japanese. Most of Chinese, Japanese, Korean men belong to Group O. A lot of Jewish men belong to Group J. Now, it's not J for Jewish, it's just an accident. But a lot of Jewish men belong to Group J. So J and the associated groups, that might be Shem's Y chromosome. Ooh, that's fascinating. Now, is that true? Don't know. But I would like to explore that more deeply over time. Okay. Now I'm going to show you a chart here. This actually makes me uncomfortable because this is a chart of skin colors that was collected by anthropologists prior to World War II. And pretty much all of the anthropologists before World War II were white Europeans, and they were incredibly racist. And what they wrote is shocking. And we would curl our toes to read what they wrote about non-European peoples, like before World War II, specifically. Still trickled in after that also, but modern, we don't talk like they talked 100 years ago. But what they did is they took a card and they looked at the skin under people's arms. with this card, and they color graded the people's skin, because that area of your skin never sees any sunlight. That actually is your skin color, is your chest area underneath your arm. That's really your skin color. And they made a map like this. And this is fascinating. But now we can apply modern genetics to this, and we learn some crazy things. For instance, you see the dark skinned people that live in southern India and in Melanesia and Australia. They have exactly the same skin color genes as the Central Africans. So, no, dark skin is not a response to living near the equator. The dark skin genes were already in the population before they spread out. Likewise, the light skin genes we see in Namibia and Southwest Africa, the same exact genetic variants we see in North Africa. Oh, because the population already had those genes before we spread out. And plus, if dark skin is a response to maybe ultraviolet light exposure, then how come the Native Americans never developed dark skin? How come the Javanese and the Borneans never developed dark skin? Because the evolutionary story of natural selection and skin color is wrong. That's just the way it is, Mr. Evolutionists. Deal with it. But something even more interesting. In African history, maybe 1500 years ago, in that time frame, there's something called the Bantu expansion. The Bantu peoples invented something called the cow. And cows are a piece of high technology. Why? Because a cow is a mobile food source. There were farmers with cows, and they're very warlike. And the Bantu people swept eastward and southward. And according to the little bit of ancient DNA we found, they killed off everyone in their path. If you went to Africa 2,000 years ago, the whole continent had a much lighter complexion than today because of warfare. Now, I'm not singling out the Africans. It also happened in Europe. It also happened in Asia. It also happened all over the Americas. I mean, people have been really bad to each other throughout time, and cultural groups and their genetics have come to dominate different places over time because of that. Okay, now let's end up with Neanderthals. We've talked about them. We had to put them in a biblical context. And this is a challenge because they are genetically different than the rest of modern humans. Fine. But we've also been deceived a little bit. You see that skeleton there? That was in the New York Museum of Natural History. Why are both of his knees bent? Because they're trying to make him look like a monkey. They're trying to make him look stupid and bent over, even though he had a perfectly modern anatomy from the hips down. Now, they do have a bell-shaped rib cage. We have a barrel-shaped rib cage. And their head, it's stuck out in the back. It's called the occipital bun, but their brains are bigger than modern man on average. They have bigger muscles and stronger bones. I mean, these are tough, robust people. Modern depictions of Neanderthals, though, look very human-like. One reason for that is because of the genetics, and we find that genes for light skin and light eyes and light hair in Neanderthals. Oh, yeah, okay, that sounds kind of modernish for Europeans anyway. And we realize the Neanderthal Y chromosome looks very much like the human Y chromosome. Okay, so they sound like people. The earlier evidence was all over the place. The modern stuff, the newest information on Neanderthals, put them squarely into the human camp. And the evolutionists don't want to say this because they think that we evolved from Homo erectus in Africa maybe 2 or 300,000 years ago. And Neanderthals split off from us maybe 900,000 years ago. They did not go through the Homo sapiens speciation event. And yet, when we left Africa, we intermarried with the Neanderthals to the point where I've got 3% of my genome is Neanderthal. Well, how'd that happen? Well, it's because they're human beings. That's what happened. They're actually descendants of Noah. There's another group of people. They're called the Denisovans. Now, we don't have full skeletons. We just have DNA evidence and some fragmentary bones. When they pull up the DNA, we're like, whoa, wait a minute. This is a whole new group of people. I suspect that this is Homo erectus. We don't have DNA from Homo erectus. We have skeletons. We have DNA from Denisovans. We don't have skeletons. I'm suspecting that there's overlap here. But this group of people from Eastern Asia, the Denisovans. Asians maybe have 0.3% Denisovan DNA, but there are people that live in Melanesia and the Philippines that have 7% of their genome is Denisovan. Add the 3% on average of the genome that's from the Neanderthals, 10% of their genome is, quote, non-modern human. Oh my, that's scary. Because it sounds like they're part ape man. And yet they're just as beautiful as everybody else, just as smart, just as inquisitive, just as creative. There's no evidence that those people that have high Denisovan levels are any different than anybody else. Why? Because the Denisovans are also people. Homo sapiens, descendants from Noah. Oh, if you look at the Neanderthal map, it's fascinating. Now, when you look at the colors as different genetic components, and yet over in Denisova Cave on the far eastern edge of this map, they found a girl who was half Neanderthal and half Denisovan. I mean, think if this happens in history, okay, fine, and we know what happened from genetics, but it's like searching for a needle in a haystack. You would never expect to find the individual that was half and half unless there was a lot less time, Mr. Evolutionist. But the Neanderthal part of that girl was not related to the other Neanderthals buried 3 feet away in the same cave. No, actually, the Neanderthal genetics of that girl was related to the Neanderthals of Croatia, thousands of miles away. What? Obviously, the Neanderthals were traveling across the landscape. They're exchanging mates, they were exchanging goods, but you could never walk from Croatia and head east and ever expect in a lifetime to find Denisova Cave. It's impossible. I mean, it's a pinpoint on a vast map of the earth. They must have known where each other lived. They must have had trade routes. That's the only way to explain that. Oh, there's so much more fascinating stuff about Neanderthals, but we have to leave that aside. And I'm going to bring it into a whole new area as we wrap up here. One, this area is unbelievable. It's the fact that they found DNA in dinosaurs. At least they found evidence of DNA. If you take dinosaur cells, wait, cells in dinosaur bones? Yeah, they're intact cells left in dinosaur bones because they're not millions of years old. They can stain them with stains like DAPI, which I've used in a laboratory, which stains DNA, or something that stains a histone molecule, which is a protein your DNA wraps around inside a cell. And there's evidence of DNA in dinosaur bones. So how far back can you go? Well, I don't think millions of years. I think then dinosaur bones are not millions of years old, and that's the evidence. Oh, and people are talking about cloning things like woolly mammoths. Oh yeah, there's a millions of dollar research program in Japan right now, and they're making progress. They want to clone a woolly mammoth. They're actually not going to clone the mammoth. What they've done is once they sequenced the mammoth genome, they said, that's the gene for hair. That's the gene for big tusks. That gene probably affects the shape of the skull. Let's take an elephant and genetically engineer it to look like a mammoth. Okay, nice parlor trick. The problem is that elephants aren't lab rats. It's not easy to artificially inseminate an elephant after genetically modifying an elephant egg, but they're working on it and they're making progress. And I'm going to be the first person online at the zoo on opening day to see that thing because that is going to be amazing. But it's not a mammoth, it's a genetically modified elephant. And the reason that they can't actually clone ancient things is because the DNA is all broken down, which is why Jurassic Park is completely ridiculous. Don't ever ask me to watch science fiction with you, especially a Jurassic Park movie. I can't stomach the science. It's so bad. Anyway, all that said, looking at the patterns of DNA we see, looking at the claims from the Bible that all people on earth came from Shem, Ham, and Japheth, and before that from Adam and Eve, consider these New Testament claims here from Colossians. Here there's not Greek and Jew, circumcised and uncircumcised, barbarian, Scythian, slave free, but Christ is all and in all. You see, my friends, there are no racial divisions in Christianity, which means we can go to the Negrito population of the Philippines, these short, little, very dark skinned people, and say, brother and sister, you're just like me, just as sinful, just as fallen, and just as derived from Adam and Eve and Noah. And no, you're not part caveman. Jesus loves you. You're one of his. Man, without that, the gospel might be hard to share with some people. But the Bible also says something like this in Isaiah 59. A redeemer will come to Zion, to those in Jacob who turns from transgression, declares the Lord. That's interesting because that word redeemer, that's a kinsman redeemer concept. We see that in the book of Ruth. Boaz decided that he wanted to buy a piece of property for Naomi, who had sold her property and then spent the money. It was her family property, but she couldn't get back. She didn't have any money. So Boaz was a kinsman redeemer. He bought the property and got Ruth in the bargain. Okay, that was a nice deal. Of course, it's in the lineage of Jesus, really important. But he was a kinsman redeemer. But when Isaiah talks about a kinsman redeemer, he's talking about a Jewish person. This concept in the New Testament is broadened to include all people. But how can I, who don't have a drop of Jewish blood in my body, have Jesus Christ as my kinsman redeemer? Well, the answer is I'm related to Christ, not through Abraham, through Adam. 1 Corinthians 15, thus it is written, the first man, Adam, became a living being, the last Adam became a life-giving spirit. And hallelujah, because if Christ wasn't related to Adam, you, me, most of the people in the room, I can't tell what racial stock you are, but most of the people I'm guessing in the room are in deep trouble. But no, now in Christ Jesus, according to Ephesians 2, you who once were far off, that's me and my Viking, pagan, Celtic ancestry, have been brought near by the blood of Christ. For he himself is our peace, who has made us both one, and has broken down in his flesh a dividing wall of hostility. I don't know about you, but I'm quite excited about that. And the fact that ancient DNA points us in that direction is amazing. So, things to take away. One, yes, ancient DNA really exists. Two, ancient DNA tells us of very important places in ancient history. Three, it does not contradict the Bible. In fact, the evolutionary time frame is directly contradicted by ancient DNA, not the biblical time frame. But we should expect curveballs, for this is a new field, and things like Denisovan suddenly pop up. Whoa, where'd they come from? So you can expect more big surprises in the future because this is not a 100-year-old field, it's a 10-year-old field. And everything's changing very quickly. And the more we learn, the more interesting it is. And honestly, the better it fits the biblical picture if we put on our thinking caps and think through the issues carefully. Now, if you like that, if you'd like to share this with someone else, go to creation.com. You can either order the Ancient DNA DVD, which, I know a lot of people don't have DVD players anymore, but you can also stream it right off of our website. And that's there if you want to see it again. There's other things on creation.com, like my favorite talk I've ever given. It's called the high-tech cell. Here I'm trying to compare the genome to a computer and very, very quickly realize that the genome is more sophisticated than any human computer program that anyone's even imagined, let alone written. It's very complicated, and the genome is absolutely amazing. That's there, DVD format or streaming online. Or look at Evolution's Achilles Heels. This is an award-winning documentary we made. It's an hour and a half long. We interview 15 PhD scientists for the movie and 9 for the book. And all we did was say, okay, Mr. Scientist, who happens to believe the Bible, what can evolution not answer in your area of expertise? And they said the most incredible things. We call them Evolution's Achilles Heels. That's there for you. On a streaming site like this, I have the first one we ever put up is absolutely free. It's called The Historical Atom, Theological Conundrums and Scientific Implications. Of course, I'm talking about the Y chromosome. If you want more information on genetics, Y chromosome, that's also there for you on our website. I'm going to leave you with 1 Peter 3:15. In your hearts, honor Christ the Lord as holy, always being prepared to make a defense to anyone who asks you. For the hope that's within you, yet do it with gentleness and respect. My friends, we've got the answers on our side. We've got the information. It's telling us the Bible's true. So we can stand confidently and competently with the Bible in our hand and defend our faith to what is becoming a more aggressively anti-Christian environment. But the truth is on our side. We just have to study enough to be able to handle a little bit of it. Honestly, the random person on the street knows nothing about ancient DNA. If you know a little bit, oh man, the conversations you could have would be amazing. Okay, so I'm going to stop there. I'm going to turn it over to the hosts, and they're going to tell me if we're going to do questions and answers or if we're going to just close, because for now, 8 o'clock. Thank you so much for listening.
Thank you, Dr. Carter.
You're
That
welcome.
was excellent. And I imagine we will have some questions if you have some time.
I got time. If you just repeat the question at the microphone, I'll do my best to
All right.
to
Well, we'll
say the
take a
answer.
microphone around to people in the audience and
Okay.
they'll fire you a question.
Hey, audience, please ask me questions about science, not about theology, even though I love theology. That's not appropriate for tonight. So Bible translations, end times theology, free will versus predestination, not cool. And can you do me one more thing? please don't ask me a question about COVID-19 because it doesn't matter how I answer, someone's going to get mad. And I don't want to split the audience. And plus, I'm going to answer it not in the way you hear on social media because I am a scientist and I know statistics and I don't want to go there. So let's not do that, okay? Good? All right, who's got the first question?
I just have a question for you. I don't know if you've run into Nathaniel Jeanson before. He's got a book called Traced. I was wondering if you had heard of that and read it.
Nathaniel Jensen is a good friend of mine.
Excellent.
We've talked many, many times for hours and hours on all sorts of topics. We've worked out together on projects. We've encouraged each other. That Y chromosome family tree that I showed you, he and I built that together. So we will both have a good Y chromosome tree that we can use in our talks.
I thought it looked familiar.
I've read Traced. I reviewed Traced on creation.com. In fact, if you go to creation.com, just type in Traced in the search bar, my review will come up. There are some excellent points in it and some things that how do you say, I don't say contradicted. I criticized with a, scholarly criticism. The one thing that I flagged was an aspect of his model where he takes that tree and he looks at all the branches and says, okay, well, this branch from wherever Noah was in the middle is 4,500 years. So therefore, this is 1,000 years, 2,000 years, 3,000 years, 4,000 years, 4,500. He's claiming he can date the events that led to the branches. And I'm not a fan of that. Something I wrote a while back is an article called Patriarchal Drive. Mutations accumulate based on the age of the parents. And the Bible has really old people that are potentially having children. So if Shem has a child at 500, that child's going to have a lot of mutations. But maybe he has a son at 400 who has then a son at 100. So Shem has a child at 500 and his son has a son at 100. Well, Shem's son is going to have a whole lot more mutations than his grandson. And so the branches of the tree, especially in the middle of that tree, are not going to be equal. They're not going to be like that many mutations equals this much time because of patriarchal drive. That was really my only complaint about his book. I watched all his videos, read his book. I think we're still friends, so yes.
One more thing then. You said on African DNA you do not have a lot as far as ancient DNA goes. You had said later on that a bog is a good place to find something like that. Are you planning on doing anything around Africa like that?
Well, even African bogs, they're not going to be, 5 degrees above freezing for 1,000 years. It's hot at the equator. Their swamps are not cold water. But as we dig more in Africa, they will find more. So they found some rock shelters. The Ethiopian highlands are a good place to look. There are just various places scattered around. And as we're getting better with ancient DNA technology, we're able to pull more DNA out of places where we couldn't expect. So we will get more, just Africa's lagging behind. And another reason for that is most of the DNA labs are in Europe. This is why the sequencing efforts were on Europeans first, and why the ancient DNA was starting to be found in Europe first. Well, it'll get there.
Dr. Demick, we have another question. We'll get him a microphone.
Okay.
If you don't mind, I'll ask a question that comes from your DVD, Spectacular Coral Reefs.
Okay.
I'll start. Are history questions okay?
I guess so, especially if dealing with coral reefs, since that is my PhD theoretically.
Oh, well, you said something, just a brief allusion in Spectacular Coral Reefs that wasn't about coral reefs,
Okay.
but I thought it was absolutely tantalizing. And you quoted a late 1800s creationist scientist, the Duke of Argyll, I believe.
Oh, yes. Go ahead.
As saying, as implying that there had been heavy coercion involved
Yes.
in the acceptance of Darwin.
Yes, if you go to creation.com and type in Argyll or Duke of Argyll, the reference will come up and you can read that essay today online. And it's a wonderful essay, but go ahead.
Oh, good. I'm glad to hear that because I tried to, Google the Duke of Argyll. Robert Campbell, was that his name?
Yeah, I think it was, yes.
Yeah, his writings are very dense and, and hard to get through.
Yes.
So thank you for doing all that work for me. I will go and do that. And you said again, what's the key
Just,
word?
just go to creation.com
Uh-huh.
and type in Duke of Argyll,
Duke of Argyll. Okay.
and then look for references. There'll be, because I know I've written in at least one or two of my articles, so I know you can find it there. But for the audience, the Duke of Argyll was a political commentator in the late 1800s, and he wrote about his phrase, a reign of terror at the hands of the early Darwinists, as they were muscling their people into all the important positions in religion, academia, and politics. And it was really shocking language that he uses. And then he uses an example from the coral reef world, how Darwin was absolutely wrong about how coral reefs form, and how they've known it for decades, and yet they resisted this, even though Darwin was completely wrong, because you can't fault Saint Darwin for anything. And if, as they're trying to promote early versions of Darwinism, it would not have looked good if Darwin was fantastically wrong on one very important subject. And that was his examples that he used for the reign of terror.
Okay. Okay, thank you for connecting those dots. It is of particular importance to me because as an undergrad, I had read the Genesis flood. It wasn't very long since it came out, but I encountered a very persuasive professor who persuaded me, you can't trust those young earth creationists because they are biased, they're trying to prove the Bible. But on the other hand, the guys who gave us the long ages and evolution, they were just following the science. They were following the evidence where it led. And
the number of abuses of science from that era. I mean, just my article on August Weismann. He's a good guy. Ernst Haeckel. Ernst Haeckel. He's probably the lyingest scientist of all time. He was called Darwin's bulldog on the continent. The guy was a scam artist, and his stuff. He's the guy who gave us the embryo diagrams that we still use in some textbooks today. I mean, absolutely fraudulent the whole way through, multiple cases, and he was probably the most influential Darwinist besides Darwin for promoting Darwinism. Just saying.
Yeah. Well, thank you for pulling some of the cover off of that.
You're welcome.
Is this on? Yeah, there we go. Dr. Carter, I'm just a little bit interested. I have two special needs sons, and so genetics is a little bit interesting to me, not that I understand it a whole lot. But is there a frequency increase in the amount of genetic, maybe I should say gene mutations? Is it getting more and more frequent?
The answer to the question is there is not a recent modern uptick in the mutation rate. What we're dealing with is thousands of years of mutation accumulations, which are just popping up. See, in the past, a lot of special needs people wouldn't have made it, and we wouldn't have seen them in the population, or they were there, but they were shoved off to the side because people in polite society didn't want to see someone who had issues. But also, our environment is radically different than it used to be. I mean, consider that Europeans, I'm assuming most Europeans, I don't know, because I can't see you. I know my European ancestry, we did not have chocolate, coffee, potatoes, high gluten wheat. We didn't have soybeans. We didn't have corn. I mean, most of the foods I consume are not anything that my ancestors adapted to over thousands of years in Europe. And that's kind of weird, thinking that, maybe our immune systems have been tuned to work with certain foods because your alimentary canal is a giant part of your immune system. Not only that, we don't get dirty anymore. So our immune systems are tuned to react to mycobacterium, tuberculosis, leprosy, botulism, not the microbacteria, but that we're tuned to react to certain organisms we've run into a lot in the dirt that we used to be surrounded in, and now we're clean. And so, what do our immune systems do? They got to attack something. And I think that might be contributing to like various things, but no, it's not. People ask me, is this the evidence of genetic entropy? Are we getting worse? Really, is autism going up? Is diabetes going up? And no, it's not because of genetic entropy from the last 100 years. It's because of genetic entropy over thousands of years, and now we can diagnose it better. Most things are more survivable than they used to be, and people are talking about it where they didn't want to talk about it before. So that's the rate seems to have gone up, but I really don't think it has.
So then,
Except for diabetes, because of high fructose corn syrup, which is poison.
so then, our environment and our reaction to our environment these days has uh, given us the ability to survive better. What about pre-flood? Then, do you think that there was less frequency of mutations before flood, or I'm guessing you have to guess at that, but what is your thoughts?
Um, if I were to guess, I would say that the mutation rate before the flood was lower. The environment was different. The foods we were eating was different. We also had a robust genome. There are families that have a much higher mutation rate than other families because they have a defective DNA repair enzyme. There's a lot of different DNA repair enzymes. You get a defect to any one of those, your mutation rate is going to go up. So I suspect there weren't defective DNA repair systems because God wouldn't have created any initially. And then through time, through the effects of inbreeding, which expresses recessive genes, mutation accumulation, if the creationists are right, Russ Humphreys and those guys, John Baumgartner, that there was a burst of radioactive decay specific amongst the alpha emitters around the time of the flood. Well, Noah and his family might have been showered with alpha particles, which might have affected their DNA. I don't know. I'm just thinking outside the box. But after the flood, with all the inbreeding we see the lifespan just goes shoop after the flood, and probably because of mutation accumulation. But I can only speculate.
Another question going back to Y chromosome, kind of the traced idea, but the concept of reconstructing human history through genetics, how well can that be done? Can history actually be sort of a way to set the clock for genetics by looking at patterns of migrations, of wars, of just various things that are known, and then corresponding those to a population shift, for example. Is that doable?
Do you see my family tree on the screen? I went
Sure.
backwards in my
Yep.
talk.
Yep. We do.
It's absolutely doable. It's you can look at Y chromosome patterns, mitochondrial patterns, the rest of the genome also, and you can see shifts in genetics archaeologically. You can see, waves of people moving in. When they sequenced a whole bunch of Y chromosomes from Lebanon, they found European Y chromosomes. Yeah, that's a relic of the Crusader era. They found Arabic Y chromosomes. The Arabs aren't from Lebanon, they're from Arabia. They found them there also. And then they found, Semitic, if you would, Y chromosomes. So we can see these things all over the place. And it's never endingly fascinating. I read papers every week about, some people group, and they're looking at their DNA. I do a lot of YouTube videos, but some of them are just kind of nonsense. It's hard to find a good one. Talking about, where people came from. They got to the point where for a lot of the Central Pacific islands, they can tell which island was settled from which other island. So they can trace this circuitous map of, oh, they found this island, and then went back over here and found this island, and they went over there and found that island. And you can actually map out the flow of the people across the Pacific to all these islands. And that's amazing. I mean, absolutely fascinating. We know that the first people that moved across Pacific, they came from Taiwan. They came from the Austronesian speaking peoples that live in the highlands of Taiwan. That's weird. Or Formosa in the ancient times. Um, and yet in the Pacific, after that first settlement, there was another wave of people, the Melanesians, darker skin, but some of them made it pretty far out. And then another wave of people came through. So we see pulses of genes in the archaeological genetic record. And I'm literally, this talk could have been 3 hours long because there's so much to talk about, but I only gave you a couple of examples.
Well, I think that's all our questions, Dr. Carter. So thank you for your time and your expertise. This was fascinating. So I think,
my pleasure.
Yes. That was a round of applause. We're gonna close with a word of prayer. Again, dear Heavenly Father, dear Creator God, we thank you for this opportunity to learn more of your amazing creation. Your creative genius and power are overwhelming to our human minds. So thank you for scientists who are willing to acknowledge your hand in everything we see. Be with us as we go from here and open our eyes to see your wonders all around us. Amen.
Thank you, everybody. Bye.
Good night.